Hello students, welcome to our lecture. Today we're going to be talking about chromosome structure and chromosome abnormalities. So in a previous lecture, we had already discussed some types of single gene disorders. So we have also are going to look at in another lecture disorders that are more related to multiple genes and interactions with things such as the environment as well. Let's take a shift now and let's look at a little bit more of a macro level for a moment, and let's talk about what type of abnormalities and disorders present themselves when we're talking about chromosome structure or chromosome number. So we're going to do a couple of different things. Let's review chromosomes first. It's been a little while since we've talked about them. So we're going to review chromosomes first. We're going to talk about the anatomy and basically structure of a chromosome. We're going to use some chromosome shorthand so you can sort of understand the different numbers and parts and how we denote things. Then we're going to talk about this concept of karyotyping and what we can learn from karyotypes. And then we'll go into specifically some cytogenetics. So cytogenetics is the field of genetics that we use to describe when we are specifically studying syndromes, conditions, and abnormalities related to chromosomes. So we will do that. Okay, so let's talk about chromosomes first. So chromosomes are, once again, DNA that has been essentially bound up so that it's ready to divide during cell division. So remember, we saw chromosomes being visible when we went through the processes of mitosis and meiosis. So other than that, the DNA is loose, it's uncoiled, it's available for normal cellular function, including things like protein synthesis, for example. So that's when it's in its unbound form. When it's in its bound form, we can see these tightly coiled chromosomes. So the interesting thing is that not all chromosomes are the same. They come in different sizes, they come in different shapes, and they also have a different number of genes on them. So if you remember, human somatic cells, all of our non-sex cells, contain 46 chromosomes. Remember, that's a diploid number. You got half from your mom, half from your dad. 22 of those are referred to as autosomes, right? And then we also have one pair of what are called the sex chromosomes, which denote XX for female sex and XY for male sex. So what does a chromosome look like? If you were to look at one, which I'll show you in a picture of in just a moment, we know that the centromere is the largest constriction of the chromosome. So it's the point at which, if you remember, the spindle fibers would attach during cell division, and then the spindle fibers would sort of pull the centromere apart in order to pull both the sister chromatids apart from each other. And again, it has an important role in cell division. The centromere also divides the chromosomes into what are called arms. Okay, so you can think of a long arm or a short arm. It's a way that we can refer to different parts of a chromosome's anatomy. We also have telomeres. Telomeres are found at the tips of chromosomes, and it is a repetition of the specific sequence TTAGGG. So over and over and over and over and over again. Every time a chromosome replicates, every time it replicates and then goes through the process of mitotic division, the telomeres, the little caps at the end of the chromosomes, they get a little bit shorter every single time. And so it's one way that we know how old like a chromosome is we can tell by how short the telomeres are. Shorter telomeres means a chromosome has gone through the process of replication and division more often than longer telomeres. So here's the anatomy of a chromosome. So again, we have a picture of the chromosome under an electron microscope. This is what it would look like. And then we have a picture of a chromosome right here. So just a drawing. So here's the centromere that we were talking about. Here are the telomeres that we were discussing. We have different staining regions called heterochromatin and euchromatin, and I'll talk about that more when we talk about karyotypes. And then whatever is sort of at the top part of the chromosome is referred to as a P arm, and the bottom part of the chromosome is referred to as a Q arm. So that's just a little bit of structural anatomy for you, if you will, of the chromosome. So the centromere position is one physical feature of chromosomes. That centromere doesn't have to necessarily be right in the middle. If it is, we call it metacentric. If there's one long arm and one short arm, we would call it submetacentric. If it pinches off a little bit of a small material on one end, it's referred to as acrocentric. And telocentric is if there's only one arm in general, and we don't have any telocentric chromosomes in humans. So this would what a picture of this would look like. And remember, these are replicated chromosomes here. So again, you have telocentric, acrocentric, submetacentric, and metacentric. So again, essentially it's just telling you where the centromere is located. And it makes a difference then on the lengths of the arms, the P arm and the Q arm of our chromosomes. All right, so now that we know all about what chromosomes look like, how do we actually use this information for something that's medically important, right? Because that's what it all comes back to, is this is a tool for medical analysis, and we use something called a karyotype. So in a karyotype, we can take a person's replicated chromosomes when they're nice and condensed, and we can take a snapshot of them. We can take a picture. And what we do is we order the chromosomes in size from smallest to largest, and we can stain them with different dyes, and different parts of the chromosome becomes visible to us. So the parts of the chromosomes that take up a lot of dye are dark bands, and they consist of something called heterochromatin. So these are highly repetitive sequences that we see over and over and over again. Not a lot of protein encoding sequences are in heterochromatin. Mostly those are found in euchromatin. So these are the light bands, and you have more of those protein encoding sequences. And again, we take our chromosomes, we number them from largest. I'm sorry, I said smallest to largest earlier. We number them from largest to smallest. My apologies. Chromosome number 1 is actually the largest chromosome. And each chromosome, again, is paired with its homologue, its exact match in size and shape. So remember, you get 2 copies, 1 copy of chromosome 1 from your mom, 1 copy of chromosome 1 from your dad. We put those together when we're looking at a karyotype. So here's what a karyotype would look like. So we have our chromosomes here. There's chromosome 1, 2, 3, and again, some of them are sort of folded and bent and sort of look a little bit like they're almost waving little flags in this case, but chromosome number 1 is the longest. Technically chromosome 21 is the shortest, but it's slightly put in a position. It's in front of chromosome 22, but that's another story. And then there are the sex chromosomes. So we have 2 of the same, so this would indicate that this is a karyotype of someone of the female sex because they have 2 of those X chromosomes. So great, we have a snapshot, we have a picture. What can we do with it? Here's what we can do. We can actually have a whole subdiscipline within genetics that focuses on these variations in chromosomes. And we can look at both an abnormal number of copies of the chromosomes, and we can also look at abnormal structures of chromosomes. So if you don't have 46 chromosomes, if you have 45 chromosomes, or if you have 47 chromosomes, there are abnormalities that are related to that. So one of the ones that's best known is Down syndrome. So Down syndrome is having an extra chromosome number 21. And so that is something that is very visible when we're looking at a karyotype. You can tell that there's an extra copy of that chromosome where there shouldn't be one. So we can use them for other things, not just medical diagnostic studies. We can use them for family studies. So if we have someone who has a structural chromosomal abnormality, it was likely potentially inherited from a relative who also had a structural abnormality in that chromosome. And so we can identify relatives this way. We can look at population studies. We can look at the effects of, let's say, an environmental toxin on health. We can examine whether or not children that are being born to parents who live in certain areas that are exposed to pollution, perhaps their offspring, their children have some of these structural abnormalities. We can also look at evolutionary relationships, the banding patterns that we see, the heterochromatin and euchromatin that I mentioned. They're very species specific. However, you can also tell a lot about how related two species are by looking and comparing those banding patterns and seeing how closely they match up. So we can talk about abnormalities, chromosome abnormalities, in two ways. One is in number and two is in structure. And abnormal chromosomes account for at least 50% of spontaneous abortions. Spontaneous abortions means miscarriages. So when a woman miscarries, at least half of the time it's because something is wrong with the chromosomes. There's some sort of abnormality there. And due to improved technology, more and more people are being diagnosed with chromosomal abnormalities. So people in the past, maybe even 50 years ago, who had some sort of condition, but people didn't know what it was. So they might call them odd or strange or other sort of derogatory terms that have previously been used to describe people. Now we can actually diagnose these conditions and we can see, oh, maybe these things are different in this person because of something in their chromosomes. So let's talk about some structural abnormalities really quick here. You can have a deletion where you have breaks in a chromosome, pieces of the material are lost, and the chromosome becomes shorter than it should be. You can also have a duplication. So you can have a region of a chromosome that was copied twice, and that duplicated genetic material is inserted then, right, copied right after it, and it makes the chromosome larger than it should be. So speaking of sort of these structural abnormalities, deletions, and duplications, the larger the area that we have as far as many number of genes that we have being copied, the more likely it is that we're going to see some sort of consequence of that. And I just noticed, and I apologize for this, this image is using a term that I don't particularly care for, mental retardation. I apologize. I don't like that term. We should call it, mental differences, but I don't like that word. So again, I will apologize to you from the outset for that. I will go back and modify this lecture because I don't particularly care for that term. But in any case, if we are only copying a very small area, then it's probably, there's going to be no associated symptoms. Maybe potentially even 2 genes, there might not be any symptoms, but when you're talking about 5, 6, 7, or 8 genes and those areas are being copied, then you might see some sort of associated conditions or behaviors. In addition, we can also have something called a translocation. So this is when 2 non-homologous chromosomes exchange parts. So crossing over was an important part of meiosis to introduce genetic diversity, but crossing over happened with homologous chromosomes. So chromosome 1 and 1 in mom and dad would switch parts. What if chromosome 1 in dad and chromosome 10 in mom switched parts? You're not supposed to do that, and that's a translocation. And then an inversion is when you have a breakage, but rather than being inserted the way that it was supposed to be inserted, it gets flipped upside down. So that's called an inversion. The other type of abnormalities we see are abnormalities in chromosome number. One type is called a polyploidy. If you have a polyploid individual, this is an entire extra set of chromosomes. This can happen in plants, but it cannot happen in specifically mammals. We are not able to handle an extra set of chromosomes, and so these would cause, again, spontaneous, miscarriages or stillbirths because this is not a survivable condition. You cannot have an extra set of chromosomes and be able to function normally. And again, this happens when you have an error in cell division. It's called non-disjunction. And I'll talk about a little bit non-disjunction in just a minute. Other than polyploids, you can also have aneuploids. So an aneuploid is when you have a chromosome, an extra, or it's missing a chromosome. And it's a monosomy if you have a chromosome that's absent, and it's a trisomy if you have an extra chromosome. And this, again, is caused by non-disjunction during meiosis. For the most part, trisomies and monosomies produce inviable embryos, which again, that means that embryo cannot produce a live birth child that will then be able to survive. You just can't have extra chromosomes or can't be missing chromosomes for the most part. However, there are some conditions which are survivable, typically associated with things like an extra sex chromosome or missing a sex chromosome. And there's really only trisomy 21, which is Down syndrome, which is your survivable condition with an extra autosome. So here's what happens. You have something called non-disjunction, and this is when the chromosomes are not separating during meiosis. And it can either happen during meiosis 1 or it can happen during meiosis 2. It just sort of depends on when the non-disjunction occurs. So let's say you had a non-disjunction at meiosis 1. What would happen is that as you are creating, in this case, we're creating spermatocytes. In this case of creating spermatocytes, we're going to have 2 sperm cells that instead of having 1 copy of a chromosome, they have 2. And so you would create, in this sense, 2 potential, if they fertilize an egg, you're going to have 2 eggs that would have a trisomy and 2 eggs that would have a monosomy. This would happen in non-disjunction at meiosis 1. At meiosis 2, it looks a little bit different. In this case, you would have, of the 4 zygotes that are created after fertilization, 2 of them would be fine. Okay, so this is euploid. Euploid means what we should see, 1 male and 1 female chromosome. And then in this case, with non-disjunction happening at meiosis 2, you only have these 2 spermatocytes that are affected. And so here you would have the trisomy and the monosomy. Some examples of different types. Oh, I'm sorry, not examples. These are just some definitions. This is a really nice table that sort of summarizes all the definitions that we talked about: polyploidy, aneuploidy, and then deletions, duplications, translocations, and inversions. So this is a nice, helpful reference for all of those things we've talked about. Here's where I was coming to. In order to describe these conditions, we use something called chromosome shorthand. Chromosome shorthand is just a way to say how many chromosomes we have, and then what the sex chromosomes are of that individual, so that we can denote whether they are someone who would have a quote-unquote complete or normal set of chromosomes, and someone who are going to have an incomplete or abnormal set of chromosomes. So 46XY would be an individual with male sex. 46XX would be an individual with a female sex. 45 is someone who's missing a chromosome, and in this case you have a missing X chromosome. That's called Turner syndrome. And then here are some other syndromes that are related to that. You can have an extra X or an extra Y chromosome and have Klinefelter or Jacobs syndromes. So again, these individuals have 47. And then we can talk about individuals who are missing parts of chromosomes, or even who have translocations, for example. So this individual has a deletion on the long arm of chromosome number 7. You don't have to worry a whole lot about something complicated like this example right here, but you should be able to identify the difference between an individual who has a complete set of chromosomes and someone who has some type of aneuploid condition. So we talked about this. This particular is Down syndrome. So this affects about 1 in 1,000 newborns. It's a trisomy for 21. It also could be caused by a translocation between chromosomes 14 and 21. Again, this term is here, and I'm just realizing that this term is here, and that's why I need to update these slides. I don't like the use of that term, but so there are some reduced mental capacities for these individuals, heart defects, potential facial features that are different. These people tend to, Down syndrome, folks who live with Down syndrome tend to have shorter lifespans. Typically it happens because of the heart defects that they have, and that's why they don't necessarily live as long. And there's a higher rate in older pregnant women. So what happens is we have a higher rate of non-disjunction that is occurring. And so that's why the chance goes from about 1 in 1,000 before the age of 40 to about 1 in 100 after the age of 40. All right, so that sort of finishes up things for this particular topic. We talked about the different anatomical features of chromosomes. We talked about aneuploid conditions, including trisomies and monosomies. So you should know chromosomal structural abnormalities, and you should also know how to use shorthand to abbreviate those abnormalities.